Okay, our final speaker today is Jerome Beetz, who's coming to us from also from the University of Würzburg. Jerome received his bachelor's and master's in Dr. Holmberg's lab, studying orientation systems in desert locusts. He received his PhD with Professor Manfred Kozel at the University of Frankfurt, looking at processing of natural sounds in fruit-eating bats. He's currently a postdoc with Dr. Basil El-Youndi at the University of Würzburg, studying migration in monarch butterflies. Jerome, it's all yours.
Can everybody hear me? So great. So first of all, I would like to thank the organizers, Melissa, Sarah, and Miriam, for organizing this great symposium and giving us the chance of presenting our research today. And I feel really honored to receive this year's Young Investigator Award. So as you can see already from my title, I am also interested in the neuroethology of spatial orientation behavior. And one of the main reasons why I'm so interested in that topic could already be answered when you look back at today's talks. So it's mostly the diversity that interests me a lot. Diversity in the sense of different animals show extremely elaborate orientation skills, but also with respect to the sensory modalities that they use for proper orientation. This is the reason why I'm working in that field. And not only these animals that we saw today are of great interest for us neuroethologists, we have plenty more other animals that are well represented in our society that show elaborate orientation skills. And we also saw today that orientation behavior is not only about moving from point A to point B. It's also about flight control and to avoid obstacles while flying. And therefore, I just want to show you a small video here of this hawk that orients through the forest with such a high speed. And I'm always surprised when I watch this video, how this bird can actually succeed orienting under these circumstances. And now we already heard today, there are also animals that can perform the same task in complete darkness, namely the bats. And this is one example. This is Carolia perspicillata, the fruit-eating bat. And it lives in rainforests in South America in highly cluttered environments. And they perform orientation behavior with their by using their ears instead of their eyes. And one parameter that is extremely important to measure accurately during this task is to measure the distance to surrounding obstacles. And this is done by the bats with the help of their biosonar. Thanks to the previous talk, I just have to summarize it very briefly now. So what the bats do for calculating distances, they broadcast an echolocation call that gets reflected by an object, in this case, that is 2 meters away. And the echo needs to travel approximately the same distance back to the animal's ears. That means overall, both acoustic signals need to cover a total range of 4 meters. Since sounds travels with sound velocity through the air, it takes a particular time, in this case 11.6 milliseconds. This is also called the echo delay, from the time duration from call onset to echo arrival. If the object is closer to the bat, in this case just 1 meter, the duration that both signals need to reach the bat's ears are shortened, in this case 5.8 milliseconds. So in other words, bats just need to measure this echo delay very accurately and translate it into distance information. We already heard today that bats need to broadcast echolocation calls in form of sequences. The reason why this is that each call echo pair just only represents a snapshot of the environment and therefore carries only one particular distance. And I wondered during my PhD at the University of Frankfurt, how does the sequence character affect the precision of distance processing in these beds? To answer that question, I first of all need to record an echolocation sequence, and herefore I use the pendulum paradigm. The bed is placed in this pendulum, and when you swing the beds towards a well-reflecting target, they start to echolocate, and echolocation calls together with echoes are recorded by an ultrasound sensitive microphone that travels with the bed. And to get an impression of how it would look like if we would sit in this pendulum, I just want to show you that video here from a forward swing. And a typical echolocation sequence that you can measure in that paradigm is shown here. So here we have a spectrogram where we have on the y-axis frequency, on the x-axis time, and color-coded is the relative energy at the frequency you're looking at. And each echolocation call is represented by such clear stripes. Now let's enlarge into one call echo pair, or I call it one acoustic snapshot. Here we have the pulse, the echolocation call, and about 22 milliseconds after the call, you can see very faintly an echo here. And to get an imagination of how the animals may perceive their surrounding, I translated this acoustic snapshot into a single visual snapshot, which is shown here. So now when I start this animation, every time when this arrow points to an echo, you will see the corresponding snapshot down here, and the translated visual snapshot for our world is shown here at the right-hand side. And to my surprise, this video on the right-hand side here looks pretty smoothly. That means that the sampling rate that the animal perceives with its echoes is high enough also for us humans to see the scenery smoothly. So now to investigate which influence the sequence character has on the neural tuning, I performed multi-electrode recordings from the auditory cortex of anesthetized bats while I'm presenting either the acoustic stream as it is or single acoustic snapshots beyond any sequence character. To get an impression, I just want to give you an example. Here we have schematically shown the bat's brain, and in dashed line here magnified, it's the auditory cortex. And since I recorded with 6 electrodes, we of course also see the neural activity from 6 units in this case, which is color coded. And the corresponding stimulus that I present at this particular time point is shown here as a snapshot. So first of all, I present each snapshot with half a second pause in between so that there's no sequence character at all. And what you can see is that almost each snapshot evokes a relatively strong response throughout the entire cortex. What does it mean for the behavior of the animal for distance processing? Since each acoustic snapshot carries a particular distance information, it becomes interesting that each unit responds quite equally well to each snapshot, meaning that if we want to analyze the discriminability of distances with this acoustic snapshot, it's rather poor, and it's barely imaginable how the animals may succeed an obstacle-free flight and a collision-free flight. However, what happens if we present the natural sequence as it is with its natural temporal context? We have the same recording, and now instead of having a large activity throughout the cortex, the activity is much weaker and more selective to particular call echo elements or to particular acoustic snapshots. When we look at the population data, I want to show you here these heat maps. Each line represents the neural activity of one unit in the snapshot paradigm and in the stream paradigm. And just by comparing those heat maps with each other, it becomes quite obvious that here almost the entire cortex is active, while here the units selectively respond to particular portions of my stimulus when I present them the natural temporal context. For the behavior, it means that the temporal context is extremely crucial to analyze the precision of distance processing in bats. While we have a rather poor distance discrimination in the snapshot paradigm, we may get a high performance of distance processing in the stream paradigm. And this is just one example that shows how important it is not only to use natural call echo elements, call echo pairs, but also to show the natural sequences as they are. As I told you initially, orientation behavior is not only about flight control. We also heard it today that it's very important to move from point A to point B and in which direction we have to steer. And for doing this, animals have to have a compass in their brain. In mammals, this compass lies in the hippocampal formation, which is schematically shown here in green for a mouse brain. But we also saw today that invertebrates have extremely elaborate orientation skills for which they need a compass. One very popular example is the migration behavior of the monarch butterfly. These butterflies migrate annually during fall from Northern America several thousand kilometers to their overwintering habitat in central Mexico. And one of the most fascinating aspects of this orientation is that in comparison to a mouse brain, they possess an extremely small brain and can perform this task without any difficulties. When we zoom in into their brain, we can see that here in greenish, they also have a compass, which is called the central complex. And during my postdoc at the University of Würzburg, I would like to understand how this compass works when the animal is actively engaged in orientation behavior. So how is migration encoded in the butterfly brain? To answer this, we first of all need an appropriate stimulus. We wanted to know what kind of stimulus do these butterflies mainly use during their migrations. And herefore, one of our PhD students, Miriam Franzke, performed a behavioral experiment in this flight simulator that has already been developed by Mauritzen and Frost. So when the animals are tethered here in this flight simulator, they can freely rotate around their body axis while observing the sun here. And in this video, it's a video showing the butterfly from below. It's pretty clear that these butterflies flies towards southwest, so towards Mexico. But as soon as Miriam occluded the sun and mirrored it to the opposite direction, the butterflies change their direction, the flight direction as well. When you look at the population data, you can see here that most butterflies change their direction after mirroring the sun. And afterwards, Miriam did another set of experiments that are pretty important for our question. She occluded the sun, but instead of mirroring it, she turned on a green light spot at the opposite direction. And hereby, interestingly, also all butterflies also change their direction by about 180 degrees compared to our first situation that we have here. So these results mean that monarch butterflies interpret a green light spot as a sun, and therefore this allows us to test the compass under laboratory conditions by taking the sun, in this case a green light spot, into the lab and present them to the animals. So in the lab, I also have a flight simulator. It's a little bit smaller, but here, and additionally, I have an entire stripe of LEDs that allows me to show the green light spot at any angle that I want to have. Moreover, I want not only to investigate the orientation behavior, but I would like to perform simultaneously neural recordings from the central complex. So therefore, I established tetrode recordings from the central complex. And we also need to evaluate the electrode's position. So I tried to insert my electrodes into the central complex. I've already told you, and at the tip of the electrodes, I have a fluorescent dye that allows me later on to evaluate my position of my tetrodes. And this position, this track here, we can also reconstruct in 3 dimensions and register it into the central complex. These are 3 electrode tracks, blue, yellow, and magenta, from 3 different animals. And this allows us to evaluate our electrode position. So traditionally, central complex neurons have been evaluated and analyzed in the lab by recording from restrained animals. And I wanted to know, how does the activity of the animal, if it's engaged in orientation behavior or not, how does this may change the tuning of the neurons? So my first stimulus that I want to traditionally analyze the receptive fields. So the animal sits here on the platform and I rotate a green light spot around the animal, which allows me to compute a receptive field of the units. This is exemplarily shown here for 1 unit. And here the firing rate of the neurons is plotted against the LED's position. In this case, this unit responds particularly strong if the LED is at the left side of the animal. Then I performed another experiment. I let the animal stationary fly, so it can't steer, but it's just flapping its wings. And the stimulus that I'm presenting is the same from before. This allows me to tell me what happens if the animal moves rather than it's quiescent, sitting somewhere. In this case, the receptive field doesn't change compared to the quiescent pre-flight condition. However, we found many units that were either recruited as the animal started to fly, and we found also units that lost their sensitivity when the animal was flying. So now you could think of, okay, maybe I lost here my units and these units are completely different units than before. Therefore, I also have a control, a quiescent post-flight condition, which is shown here. And this allows me to compare the quiescent post-flight with a quiescent pre-flight situation, and it looks very similar to each other. I can further evaluate if I'm recording throughout the entire experiment from the same units by looking at the spike shapes that I get. So since I'm recording with 4 electrodes, I get in each electrode a spike shape, shown here in blue for this particular unit in the quiescent pre-flight condition. When we compare now these spike shapes with the ones that I get for the 2 remaining conditions, it becomes clear that those are highly similar to each other, which further confirms that I've recorded during the entire experiment from the same unit. So obviously, the fact that the animal flies, although it's not steering at all, changes the compass in a way that some units became sensitive and others lose their sensitivity. So now the next question, and the most interesting question, is what happens if the animal can set its desired heading relative to the green LED here? And this is in the video showing you how the butterfly orients in my setup. It can freely rotate around its body axis while I'm recording from the central complex, the neural activity. And with this condition, I can also analyze the receptive fields. And we found in many units that the receptive fields seems to change as soon as the animal is able to rotate. Just by comparing visually this receptive field from the 3 remaining ones, it becomes clear that the preferred tuning of the units seem to change. When we look at the population data from 27 units, each line represents 1 unit, color-coded as the neural activity, and on the x-axis we have the LED position. When we compare this heat map with the heat map that we get for the non-rotatory flight, we can clearly see that these areas are still represented in this image here. However, for the rotatory flight, we get a rather low similarity of this activity compared to that one. And again, for the quiescent post-flight, it becomes clear that this image again has a high similarity to our initial image. This means that most units changed their receptive field because the animal is able to rotate. So now we may wonder, why do these units change their receptive field when the animal rotates? To get a hint and an answer to that question, we plotted the preferred tuning angles that you get when the animal is quiescent. Each circle here represents the preferred tuning angle of one unit. And just by looking at this graph here, it becomes quite clear that most units have their receptive field at the visual front of the animal, while the back field is largely underrepresented. And this situation changes completely if you compare this with a rotatory flight condition. Here, instead of having a 180-degree representation of the visual scenery, you now get a 360-degree orientation. So in other words, when the animal is quiescent, it has a narrower visual field than when it takes off and flies, you get a 360-degree visual field. And again, when it sits down in the quiescent state, the visual field again narrows. These examples demonstrate that the stimulus context is very important for the animals. I showed you this for the bats, that it's very important to have the right temporal context in this particular case to get an answer how detailed distance processing is. For the monarch butterflies, it was pretty important in my case that I know that the green light spot evokes the same response as the sun, so that I can discuss my results with respect to the sun compost that I receive in the lab. And we can also see that the behavioral state has tremendous effects on encoding compost information in the central complex. So last but not least, I would like to thank all the people that influenced me over the last years, which are responsible why I'm here today receiving this award. First of all, these are of course the PIs, Uwe Homburg, who was my PI during my bachelor and master thesis from the University of Marburg, Manfred Kössel and Julio Echeverria, who supervised me during my PhD at the University of Frankfurt, and Basil Alyundi, who is my current PI during my postdoc at the University of Würzburg. Also special thanks to our collaborators, Christine Merlin from Texas A&M, David Dreyer and Eric Warren from Lund University, and also I would like to thank the International Society for Neuroethology, because I'm so happy that this society exists, because it best represents my research interests, and I think I perfectly fit into this society. And of course, thank you very much for your attention and your patience of staying here so long. Thank you very much.
Wonderful. Thank you so much, Jerome. That was wonderful. Sarah, we have plenty of time for questions.
Yes, so please send on over a note to me if you have a question.
I'll ask a question to begin with, Jerome. You may have said this, and I apologize if I missed it. Why do you think that the bats equate a green dot with the sun? That was a little bit shocking to me.
The monarch butterflies.
Yeah, sorry. Mm-hmm.
Yeah. This is because when you occlude the sun and when you wait, I can just repeat that slide briefly. Maybe I was a little bit too fast. So when you occlude the sun here, and instead of showing the animals the sun a green light spot, then most butterflies rotate the steering by 180 degrees compared to the first situation. So that means, first of all, we let the butterfly fly in a natural situation, and then in this situation, and we compared if the butterflies change the direction of heading. And that's the reason, since they change the heading by 180 degrees, that we can say that the butterflies interpret the green light spot as an LED. And I also have to mention, this has already been shown from honeybees and also from dung beetles, that they use the green light spot in a similar way as the sun for orientation purposes.
Thank you.
Miriam?
So just a follow-up question, have you tested any other colors? Would they also interpret blue light spot as the sun or a UV light spot as the sun? So is it just a bright spot, or does it have to be green?
Yeah.
That's a very good question. We currently perform these experiments, and we haven't finished them yet because this year's field season was also postponed due to COVID. However, what we also want to test, of course, is if they use a UV light in a similar way as a green light spot, or if maybe they interpret UV light in the same way as honeybees do, because honeybees interpret UV light as an anti-solar hemisphere and green light as solar hemisphere, because we have a spectral gradient at the sky. However, as I said, these are still undergoing experiments, so I cannot tell you more.
Thank you.
Great. Eric?
Eric, you're muted.
Sorry.
Now we can hear you.
Yeah, thank you very much, Jerome, for a really interesting talk. I think it's really fascinating that you've got more or less a behavioral dependency on the responses of your central complex neurons. That's a remarkable result in many ways. I still don't know if I fully fathom what it means yet, but I'm sure that's going to come out at some point more clearly. But I think the fact that you've been able to demonstrate the differences between secured animals that are allowed to fly compared to secured animals that are allowed to fly, compared to completely freely flying animals, I think is a tremendously interesting result that, if nothing else, is going to make us rethink the way we do our experiments at the very least, and at least rethink how we interpret our results relative to how we do our experiments. And I think that's a really quite interesting finding. So I'm really excited about that, and I'm looking forward to seeing how that develops. So it's not really a question, it's more of a comment.
Thank you very much, Eric. Maybe I could make a brief comment to that. So we currently try to understand what kind of mechanisms are standing behind these changes in the preferred tuning. And one hypothesis, of course, is that since the animal is really turning and rotating, that idiothetic information may play an important role to flip the receptive fields. So these are still ongoing ideas, and we also perform, for example, currently platform experiments in our lab in which we rotate the butterfly in a similar way as Roy Ritzman did it with his cockroaches to analyze where these flips maybe come from.
Thank you. Peter Nairns? Oh, you're muted.
I didn't have a question, but now that you called on me, I'll ask a question. Jerome, that was a wonderful talk, as they all were. I just noticed when you showed that beautiful image of the wide visual field when the animal was flying, and then how it became narrow when the animal was not flying, stationary. Does that mean is that why it's so easy to pick up a butterfly when it's sitting on a branch?
Yeah, that's a very good question. Yeah, actually, my initial plan was to make somebody trying to catch a butterfly in this scenery. So,
Yeah.
but yeah, it's a good question, because those butterflies also may have predators. However, those butterflies are toxic, so maybe the 360-degree view is not so important to them.
Right.
But sure, I think especially for movement detection and so on, I think it's also important to them that they still have some capability of seeing what's going around if they are just quiescent. But at least we see that there's a bias. Also, we have some neurons that still have a receptive field at the back field of the visual scenery.
Are there fewer of those though when there's quiet?
Excuse me, what?
Are there fewer of those neurons, you think, when the animal is quiet?
This is what we think, and these results also go in line with results that we have from intracellular recordings, which one of our PhD students in the lab currently performs. Because with intracellular recordings, we can explicitly get an idea what kind of neuron we have. And these compass neurons also intracellularly respond most strongly to the green light spot when it's in the visual front field. So I think we still have to work more in that direction to get a clear answer why they have such a restricted view. And of course, we also have to test this behaviorally, maybe in our arena with some movements, with some looming stimuli from the back to see if they do not respond to them or not. So it's opened up a lot of questions.
Thank you very much.
You're welcome.
Morgan Nuvian.
Yeah, hi. Thanks for the great talk. I have a question, because as you see, these neurons are changing their tuning depending on what the butterfly is doing. And I really like this idea that they need to adapt to a broader field of view. But at the same time, they also need kind of a stable representation of the world around them, right? With the direction of the sun. So I'm wondering, how do you reconcile these two ideas, both the stability with this adaptation to the context?
I personally could imagine that the neurons in the central complex may have a different purpose when the animal is quiescent rather when it is flying. So for example, when the animal is flying and rotating around, there have to be neurons that encode the animal's heading very precisely to get an idea around the world and so on. We currently hypothesize that maybe the units that we are recording from in the quiescent situation may be more sensory neurons that do not represent the visual scenery by 360 degree, but maybe particular objects in the surrounding. And this may get changed, this may be changed when the animal starts to fly and actively orient. It may be also interesting to see, but it's really hard to perform these experiments. What happens if the animal rotates on the platform? So when it actually does an orientation behavior, when it's quiescent and rotating. But it's really hard to get the animals to rotate on the platform.
Great, thank you. I think that's time, Mel, is that right?
That's correct. Thank you so much, Jerome, for a wonderful talk and a great discussion. I'm sure that you all want to join me in thanking our 2020 Young Investigator Award awardees, recipients. Thank you for wonderful talks. I'm sure that, like me, you're all equally excited about the future of these scientists and the future of the Society for Neuroscience. For neuroethology and neuroscience too, for neuroethology. I think we're in very good hands. And as Peter said, we're excited about your future and seeing what comes out of your labs in the future. I'd also like to thank all the participants that have been here. It's been a wonderful showing. I don't know what the final big tally was, but thank you everyone for coming and it's great to see everyone, even if it's on a little bitty computer. And hopefully I can take everyone out for, well, not everyone out for a beer, but hopefully I can join everyone for a beer when we can see each other in the future. Thank you so much.